Hi, this poster describes our work on the structural and biochemical basis of interaction between a certain VHH fragments derived from camels and a transporter called NorC against which it was derived. NorC is a transporter found in Staph aureus and it belongs to the major facilitator superfamily, and like its homologue, it is completely embedded inside the cell membrane. We wanted to structurally elucidate the transporter through X-ray crystallography. Because of the low solvent exposed surfaces found in MFS transporters, they are extremely difficult to crystallize, even though they are easily uh, purified. And the same was observed in case of NorC, where even exhaustive crystallization attempts yielded mere spherulites, but no crystalline precipitates. To aid in its crystallization, we sought after high affinity single domain variable heavy chain antibody fragments, um, as they have successfully been used in the crystallization of various other membrane proteins. For this, we heterologously purified NorC in E. coli cells and then reconstituted them into proteolyposome and then injected them in camels in the National Research Center for Camel in Bikaner, India. We also increased the antibody titers sequentially against NorC through repeated immunizations for 6 weeks. Isolation of VHH coding mRNA from the camel's peripheral blood mononucleosis gave us a library in which we could find 4 VHHs that strongly bound to NorC as ancestral facts. These VHH were named as Indian camelid antibodies, or simply ICAPs. We proceeded with one of them, the ICAP3. Upon solving the structure, we found out that the CDR3 of this ICAP harbored a zinc coordination site instead of a conserved disulfide bond present at the same position. When we tried to engineer the disulfide in its place, the disulfides were successfully formed as confirmed through mass spectrometry analysis. We found that not only did the thermal stability of NorC was decreasing, its affinity for NorC also decreased by 100-fold in case of one disulfide forming mutant called V115C, where it was completely abolished in case of H116C mutant, which also happens to be involved in the formation of the coordination bond. It seems that the zinc was providing a tether for CDR3 to stabilize as a specific paratope against NorC. MD simulations confirmed our hypothesis, revealing a much more dynamic state of CDR3 in the absence of zinc at the coordination site. Through confocal microscopy, we tried to find out the topology of this ICAP's interaction with NorC, and as can be seen here, with NorC probed through C-terminal GFP and ICAP through a covalently bound rhodomy, it was only when NorC was expressed that ICAP was co-localizing on the E. coli spheroblasts, where the dead mutant ICAP3H116C did not. Um, Once the topology was established, we proceeded towards co-crystallization attempts, and after rigorous screening, we were finally able to reach crystals that regularly diffracted at 3.6 angstroms resolution. Phasing with selenium sat gave us an accurate map of the structure with NorC coexisting in an outward open conformation with ICAP3. The transporter looks strikingly similar to distant homologs with completely different functions as well as secondary structure topology. The ICAP seems to protrude inside the cavity of the transporter, forming numerous hydrogen bonds with many residues of NorC through CDR1, while CDR3 seems to make a near perfect surface complementarity with the transporter. This effectively blocked much of the solvent access, and hence TPP, a small molecule that binds to NorC with micromolar affinity, fails to associate with it in the presence of ICAP3. On the other hand, TPP did not seem to have an effect on the affinity of ICAP for NorC, suggesting that TPP binds in the remnant of the vestibule, somewhere here, still inaccessible to the ICAP. Most surprisingly, we found that NorC in its native environment may exist in one conformation, say the inward open conformation, and the presence of ICAP pushes it to attain another here. And this was assessed through pegmalamide accessibility assays. To summarize, we isolated ICAPs from camels immunized against NorC. These ICAPs helped in crystallizing NorC, which yielded a near atomic resolution structure of the complex. The ICAP blocks solvent access to the function site and also altering the conformation landscape of the transporter in its native environment. Thanks.